Hello.
Hi, say some more.
Hey, yeah.
I didn't expect you to come. How are you doing?
Doing good. How about you?
Doing great too. What's new with you?
Nothing, just regular stuff.
Okay, nice. You're not taking DLH, right?
No, I already took that.
Oh, I see. I see.
But I like the reading group, so
Thanks. That's great.
papers and
Cool. How did you like it when you took it? What was hard about it?
everything was hard about it. So actually, I timed it wrongly. So that was my second course, and so I didn't know anything. It was first time offered.
I see.
I got greedy. I thought, I don't know what will happen. Maybe they'll not offer it next time.
Oh, so this was last year? Last year was the first time it was offered?
Yeah.
Ah, interesting. I didn't know this is the second time. That's pretty brilliant. Huh.
So, but it's I mean, the content is little dry. I mean, honestly, you have to put in a little, if you want more insights. It covers the width, so it will introduce a lot of good architectures. But probably if you want to do some deep dive, maybe some input is required. I mean, some more effort is required from your side.
I see. I see.
Assignments will be hard.
Yeah, assignments are fun so far. The first one is.
It will be hard. I mean, don't look at the first. I think the first assignment was tough for people, but going forward, there will be few. I mean, I remember. I mean, almost everyone will be tough.
Yeah. Hi. How do we say your name? Can
you can say Virendra. So
Veer? Okay, nice to meet you. Ilya, in DLH this semester. I think Samarth joined us for NLP Open Reading group last semester, but I don't think he is taking DLH this semester, but he has already taken it. We were just talking about it. Okay, wow, okay, people are coming slowly. Let's see. I know Rahul is very committed to coming almost always, and I see he's online, so I assume he will also be coming.
Hi everyone, this is Sandeep.
Hi.
Yeah, I took DLH last semester, so I just joined this group. Yeah,
gotcha.
So just
so you're like you must have taken it with Samarth. He also took it last semester. Last time it was offered. I'm just waiting for maybe two more people to come, because I know there were more people planning to come today. We could start.
Yeah, did you change your apartment?
I'm staying at a friend's place.
Yeah, okay. Yeah, I like it also. The old background was also good.
Hi, Gil.
Hey, how are you?
Doing great. How about yourself?
Same here, same here. Hello to everyone else.
I know Raj is also coming today, but he's a little late. I think we can start maybe just, okay, so the general idea of these meetings is, the last semester we did the test trial with the NLP class, and I personally got a lot out of these meetings. I remember very well the papers I presented, and much better than I would have remembered them without presenting. So at the very least, it helps me remember things I present. And I think the same is the case for other people, but also just hanging out with people, talking about class-related topics is fun, and gives you some sense of community. So this is kind of the sorts of things I personally want to get out of this, but it's also a very malleable thing where everyone is open to suggest format changes, et cetera, So, and I would like to suggest that everyone does a quick personal introduction. I'm Ilya. I went to UIUC for my undergrad, but originally I'm from Russia. Right now I live in Lisbon, Portugal. I work at Cloudflare as a systems engineer. My work involves a lot of like Linux kernel networking and systems stuff. So the thing I'm trying to get out of the MCS classes is learn about things we don't work with at all in my day job, which is like all those deep learning things that I want to get some exposure to, because I'm not getting exposure to them in my work. So yeah, that's a bit about me. What about other folks here?
Yeah, so I'm Akhil, and I say quite similar what I want to say as Ilya said. I am in London, UK. I work in finance, basically kind of algo platform. I developed that, so that's kind of everyday stuff. And I'm more interested in learning, kind of as you mentioned, like deep learning and the neural network and that kind of stuff. That's why I started this class. So yeah, same like skill wise, I'm very excited to learn new things about whatever language, whatever things, whatever tools. Yeah, just like that.
Hello, everyone. Nagin here. I just joined. This is my first session.
What classes are you taking? Or you graduated, right?
Yes, I'm in.
Have you taken DA before?
Have I, what, sorry?
Have you taken deep learning for healthcare?
No, unfortunately I couldn't, but because it was introduced to the program recently, if I'm not mistaken, and they offer it only in spring, and I had other classes in spring, so I couldn't take it. But I'm excited about this group and the whole process that we're doing.
Cool. Welcome.
Thank you.
Dear, what about you?
Yeah, can I go next?
What is it?
Yeah, sure. Yes.
Okay. I hope I'm audible enough.
Yep.
Yeah, hi. So let me introduce myself. So I'm Virendra. So I have enrolled for USC program 2 years back. So I completed 6 courses so far. I have 2 more to go, which is deep learning and ABM. Yeah, so it was a kind of good journey so far, so many learnings. So professionally, I work for Oracle, so I've been into the application development, the Fusion application development. We deal with a lot of data, but in the current role or all the journey that I had with Oracle, it's more of applications development. So I'm trying to get exposure to the data side of it and drive the business based on the data and what we gather from the insights that we gather from the data. Cool. Yeah, so I'm very excited to hear about this group. I wish we had this kind of initiative much long back, in the sense that we had started taking the other courses as well. Yeah, but yeah, I really look forward for more learning as well as contributions. Yep. mindset.
Great, perfect. It's fantastic. Samarth.
Samarth has got beard this time.
Yeah, I'm trying to
like
get away with cold. This time it was too cold. So hi everyone, this is Samarth. I joined this course in fall of 2020. So till now completed 4 courses. DLH was my second course, and it was introduced first time when I took it. So it's an amazing course. And I was part of the reading group what Ilya started in NLP as well. So that is even better, I would say. You learn a lot. Regarding from professionally, I work with SAP labs here in San Francisco Bay Area, and I'm a data scientist there. Work with building these kind of models for enterprise applications. So really excited. I mean, I love this topic, so couldn't resist from joining again. And thanks, Ilya, again for hosting it.
That's great. Sandeep, what about you?
Yeah. Yeah. So I started the course in 2020 summer, and this is my last semester. I'm taking ABM. I took deep learning last spring, and I currently work in healthcare domain, work in integrations using REST applications with other tools, integration tools, MuleSoft, SOA. We use all those integration tools, and we do some visualization of in the healthcare. We have Tableau, those applications, but not data science or machine learning applications. So yeah, out of this course, I was looking at exposure to machine learning, data science, so and even deep learning. So that's what I was looking to get out of this course. And previously I've worked on automotive industry, retail, and other industries. So yeah,
I think among all of us who probably know the most about the healthcare industry, so you'll be calling out people's mistakes, and especially mine. Rahul, welcome back. Rahul and I took some courses the last semester. Want to introduce yourself?
Hey everybody, Rahul Singhal here in Chicago. Sounds like we're just giving a little background info. So I joined the program last semester. As Ilya mentioned, we were in NLP and 410, and I was also taking cloud networking last semester, full-time student. So I have at that time, he was a 4-month-old baby. So he's now 9, almost 10 months old. And so I was on night duty, so I had a lot of late nights studying, and so which is why I'm late, because I was up all night with him as well. So my background is in there's a variety of things: insurance, e-commerce, enterprise tech. I did sales for a while. GC'd the house I live in right now. So this program is sort of a return to home of getting back to hardcore CS, as well as the machine learning world. I did a lot of this stuff actually back in high school and undergrad at U of I, but the algorithms were terrible, the hardware was terrible, and MNIST, I think, was the only public data set at that time. So this is very exciting for me to get into the healthcare. Well, healthcare I'm sort of okay with, but definitely getting into all the deep learning models and seeing how they apply. So excited to be here with you guys and learning from all of you and share whatever insights I can.
Perfect. Okie dokie. Okay, I'm gonna start sharing my screen then. So I only have one screen today. So, okay. Look, Zoom does this overlay, I guess. Can you folks see my screen? Okay, great.
Yes, I can see.
I might not see if you visually try to indicate that you say something. So just speak up and interrupt me if you have any thoughts or questions or you think I'm wrong, which is likely. Okay. Yeah, so the paper I wanted to talk about today, let me adjust things a little bit. Okay. Yeah, the paper I wanted to talk about today is, do we even have it open? I don't think I have it open. Oh, okay, it's right here. It's called Semi-supervised learning of the electronic health record for phenotype stratification. And basically this was published in 2016 and it uses some of the some deep learning health techniques, which might not be the most up-to-date ones today. But the reason it's an interesting paper is because it very nicely introduces you to the concept of electronic health records and what you can do with them and what sorts of problems you have in there when you try to analyze them and what you can get out of them and that kind of stuff. And also, this is, for me personally, the first deep learning paper that I've read with a lot of medical terms. So I have a lot of Wikipedia tabs open because while reading it, I had to do some bio reading up. Yeah, so I suggest, I'm just gonna, I don't know, have people had a chance to download this paper themselves? You folks can, if you want, open it on your own.
I have not downloaded it yet. Do you recommend that we do that now and keep it open on our own screen, or do you want us to watch you?
I think, okay, so I would like us all to read the abstract right now, to spend a minute, like everyone by themselves reading this abstract, just one paragraph. And then for the rest of the discussion, I can just share my screen. I think that would be fine.
Okay, do you want to give us a quick introduction to the terms in biology that you had to really get into?
I think after you read the abstract, then we can talk about terms in biology. So yeah.
Perfect.
So I'm just going to pause for a minute and go through the abstract, then we can resume. Okie dokie. So now I'm going to talk about the terms. So the most interesting thing, the paper itself focuses on this thing, which is very common in medical papers and medical analysis, is this genotype-phenotype association. And in particular, with data science, you can leverage EHRs to infer some association between genotypes and phenotypes. Now, what are these things, genotypes and phenotypes? Genomes you're more or less probably all familiar with, which is the entire set of genes that encode human biomedical parameters, I guess. And phenotypes, unlike genomes, phenotypes are parameters which can be visually seen in a human. All the traits which, like eye color and stuff, or hair color, that's a part of the phenotype. In medicine, apparently, certain diseases are also classified as phenotype. And it turns out that there's this concept of, so I actually have a few graphs pulled up. There is a concept of, let me back up. So a genome is represented as pairs of nucleotides, and there's 4 types of them. I don't remember their names, but basically they're coded as C or G or A or T or T. And for any human being, we have millions of genes, and most of these pairs, base pairs they're called, in a human genome are shared between humans. But then some of these might vary between humans, and some of these might be have innocuous differences, and they might define things like your eye color or some other parameter. But other variants might be causal in you having a disease of a particular type. And a classical approach in learning what part of the genome modification is related to a particular disease is in general called these single nucleotide polymorphism studies, where the idea is to try to find a correlation between changes in a single genome when a single base pair flipped, which is commonly the same among all humans, but in some person might be different, or in a couple of people it might be different. And the idea is to try to find correlations between those deviations and diseases. So if I open the single nucleotide polymorphism wiki page, I will find the word disease mentioned a lot of times, because SNPs pinpoint differences in our susceptibility to a wide range of diseases, et cetera, et cetera. So the idea of a lot of medical studies is to take the phenotypes of a human. A part of the phenotype is a disease, which we have detected, and take the genotype of a human, and then run a study trying to find correlations between the two, and then look for causal relationships. So as you might imagine, you can use EHRs, electronic health records, to automate to bootstrap the data on human phenotypes, right? Because all the parameters like your blood pressure levels or some other stuff that the doctor might examine, different vital parameters can be taken from your EHRs and can be used as a phenotype. And then if you have the genotype taken from somewhere else, you can run those studies trying to find correlations between the two. Okay, so this particular paper focused on the fact that a lot of EHRs don't have reliable data and have a lot of noise in them. And this paper used denoising autoencoders to reduce the dimensionality. So every EHR has very high dimensionality and has a lot of different parameters on the human because they regularly have tons of entries. But then different EHRs might be missing different feature values, right? You might get one doctor might measure one thing for a patient, but for another patient it will not be measured, or a certain parameter for a patient could be measured incorrectly. So the idea of denoising autoencoders is to improve the quality of data, and it's specifically very useful with EHRs. And this paper focuses on the use of denoising autoencoders on EHR data. So let's see, where are the good examples? Let me. So the idea of a denoising autoencoder is to take an input layer. Can I select here? Let me see. Oh, I have a laser pointer. That's great. Okay, so the idea of a denoising autoencoder is to take what is it? 1 second. Isn't it? Okay. Is to learn to take a corrupted input and then reconstruct the correct input. by using a representation with reduced dimensionality. So the idea is to train a small neural network with just one hidden layer, where you would take some data, and we'll talk about what data that is. But for now, let's just talk about a vector with a bunch of feature values, and corrupt some of the values in that vector. And then train a neural network with one hidden layer to reconstruct the original input. Right? Now, you can think this is a denoising autoencoder because the idea is that it autoencodes the data, in that it encodes the data in a reduced dimensionality way. Yeah, Najin, sorry if how do I pronounce your name once again?
It's Negin.
Negin, okay.
Yes, thanks for asking. Quick question, the reconstruction, what is the difference between reconstruction of the hidden layers and activation of the each unit of the layer? Because those kind of seem the same to me. You either deactivate some of the units or reconstruct a whole layer so it will have less units.
Can you ask once again, please?
So, for example, in the reconstructed layer that is written here after the hidden layer, yes, over there, there are 5 units in this hidden layer. So if we reconstruct it and then eventually it has 3 units instead of 5, that is
wait. You just said a sentence, if we reconstruct it and it has 3 units instead of 5, what does it mean?
I mean, it means that the reconstruction is only 3 neurons.
Instead of having, for example, 2 units to 5, then we can reduce the number of units.
Reduce where? In the reconstructed layer?
Yes.
But then the idea is that so the dimensionality of the correct input should be the same as the reconstructed layer, right? Because the ideal reconstructed layer is to reproduce the correct input, which you have corrupted, right? So you have the goal of this network. It's not a classifier or anything. It's the goal of this network is to get the legitimate input data, right? Then some of that data we corrupt intentionally, and the network needs to learn to reproduce the original input, which it never gets to see, to reproduce it from the corrupted data, right?
Yes, sorry, I confused that because you said the dimensionality reduction, and I thought that.
Okay. Yeah. So, yes, I was getting ahead of myself. The idea of the reason I was mentioning dimensionality reduction is that by itself, this thing is not very useful. What you do then is you train this in this way as an encoder which denoises your input, but then you take this network, or actually these 2 layers over here, and you throw this layer away. And what you do with and then you want to plug these 2 layers into an actual classifier. So, what you will do is, so this is actually shown over here. You will have your input, which will obviously, in an actual classifier task, will not go under any corruption. So you'll take that input and then you will feed it into this denoiser that you just trained before, and it will give you a lower dimensionality representation of that thing. And then you will be able to train a classifier on that lower dimensionality representation. The reason a lower dimensionality representation is desired here, and the reason we're not making the hidden layer larger, right, is because we want to apply this problem to EHRs and the biomedical records in particular, where we actually have very few labeled examples. So the constraint that the authors of this paper are facing is that they have a lot of, they can bootstrap a lot of data to train a denoiser, the autoencode, the DA, but they don't have much data to train a classifier. For example, if you want to classify a patient into a sick patient with a particular disease or a non-sick patient with a particular disease. But they still have the problem of data not being good enough and reliable enough. So what they do is they use a lot of data, a lot of simulated data in many cases to train this denoiser first, which is able to find this, learn this lower dimensional representation for input. And then since you only have 2 or 4 or 8, but not many units in this lower dimensional representation, instead of thousands of units, which would correspond to an actual raw EHR, you're then able to train a classifier on very few training examples, or not very few, but fewer than normally. Yeah.
Thank you.
Does that make sense? Yeah.
Of course.
Okay, cool. Yeah, the issue with these papers is that I never know which sentences to read out loud and which not to read out loud. Okay. So I'm gonna read this paragraph. We evaluate DAs, the denoising autoencoders, for phenotype construction using 4 simulation models of EHR data for complex phenotypes, modify DAs to effectively handle missingness in data, and use the DA to create cluster visualization that can aid in the discovery of subtypes of complex diseases. And then they apply these same methods to, so this that I just read out, they do on simulated data, they train those DAs and then they visualize their efficiency, but then they also apply the same methods to patients, LS patients, which I'm not sure what LS is, and they only spend a few paragraphs talking about patients from real true data. So we're going to skip that part and talk about the simulations that they run on TA. And you'll see what I mean by simulations in a method in a second. So let me go through this section real quick. So they give a motivation for using DAs here. So the DAs were initially introduced as a component in constructing the deep networks used in deep learning. Recently, deep learning methods have been useful to solve biological problems, including tumor classification, predicting chromatin structure, et cetera, et cetera. DAs in particular showed strong performance early in the deep learning revolution, but have been surpassed in most domains by CNNs and RNNs. While these complex deep neural networks have surpassed the performance of DAs in these areas, they rely strictly on structured relationships, such as the relative positions of pixels within an image. This structure is unlikely to exist in the EHR. In addition, complex deep networks are notoriously hard to interpret. DAs are easily generalizable, benefit from both linear and nonlinear correlation structure in the data, and contain accessible, interpretable internal nodes. Oftentimes, the hidden layer is a bottleneck, right? So this, where is the architecture? This bottleneck is introduced there on purpose. As a bottleneck, a much smaller size than the input layer, in order to force the autoencoder to learn the most important patterns in the data. We use the certain library to construct a DA consisting of 3 layers, an input layer X, single hidden layer Y, and a reconstructed layer Z. Noise was added to the input layer through a stochastic corruption process, which masks 20% of the input values. Okay. Is the general idea of the DA, does it make sense so far? And we'll see its application to the EHR records in particular in a second. Okay, cool. So the DA sort of makes sense. Cool. then what is confusing and interesting about this paper, actually, let me turn my lights on because it's getting dark. Okay, so yeah, what's confusing but also interesting about this paper is that they ran a lot of simulations with their own simulated data to understand how DAs work and whether they're useful to medical applications. And only then the researchers actually applied the DAs to whatever they wanted to solve. But most of the paper talks about these simulations that they ran, which is kind of a new approach to me. So I'm gonna go through this section real quick.
There's a couple questions.
Yeah, what's up, Rahul? Or sorry, Veer also raised hand.
Yep.
Yeah.
Yeah, I just want just trying to understand the DAs. So can DAs be, let's say, if we apply against an image, so can it be seen as like you take an image, you smudge some pixels, you erase some pixels, then
Yep.
you the DA refix it based on the boundary pixels, something like that?
Yeah. So that's how you train a DA. Yeah, that's precisely the idea. But then after training the DA, after this pre-training, if you throw away the final layer, you can plug in this intermediate representation into some other task, like a classifier, right? And because it's a demo, like you don't need the entire picture to learn the outline of a cat, right? You can do a lower dimensional representation of a cat versus not a cat if that's your classification task. Yeah, and the DA is an efficient way to learn that. Who's the next person who raised hand? I can see. You can just speak up.
Yeah. So Ilya, Samarthya, quick question. The cost function which is mentioned here in this DA, is this a special kind of cost function they have mentioned, or is it the generic cost function?
No, I don't think they did anything. So this looks like
this looks like a BCE cross entropy, a binary cross.
Yeah, that's just categorical. Yeah, cross entropy.
Cross entropy. Yeah, but essentially the reconstruction, I think cost function can be depending upon your input and output, right? So let's say if these are sensor values which are coming in, let's say as input,
yeah,
instead of okay, then you'll use a different regression, maybe MSC or some other kind of.
It averages that and learns from it, if I'm not mistaken.
Yeah, that's by entropy here. So the question is, they have used cross entropy specifically because the inputs are categorical in nature.
The outputs are the phenotype stratification. They're trying to classify into the specific phenotype.
No, but the classification is done at the end of when you feed into the classifier. But deconstruction layer cost function.
What you're asking why this cost function is picked?
Yes. So it gives me a hint that the inputs are like categorical in nature.
Why not really why cross entropy? Is that your question, right?
Oh, the categorical in nature isn't like 0 or 1. Okay.
Yeah, one hot encoded.
Oh, okay. This is a perfect segue into the next thing that will. Let me walk you through the simulation framework that they talk about, and then you'll see what they mean. Okay. So yeah, you can just bring this question once again after this section if you think that it still makes sense. We designed 4 simulation models to evaluate algorithmic performance. These simulations were not designed to perfectly recapitulate EHR data. Instead, they're designed to capture a variety of complexity in order to identify algorithmic strengths and weaknesses with known underlying models. To simulate patients with first clinical observations were generated by first drawing random samples from a normal distribution. Next, okay, so this gives us a certain feature vector, right? You draw samples from a normal distribution. Let's say you have a feature vector of length 10. Next, hidden input effects were generated in accordance with one of 4 simulation models. So you add a few more parameters to your vector, where you input, you generate hidden input effects according to one of the 4 models. And they used 4, we'll talk through those models in a second, and they looked at those models to see where DAs perform well and where they underperform. When turned on, these hidden input effects, okay, this sentence, I don't like the sentence, let's ignore it. Shifted, let's see. Okay, so here they give an example of, let me think, is it better to go through? Okay, let's talk through this first model and then look at the visualization, and then I think it should start clicking here. There are 4 models defining hidden input effect rules to determine case control status. So are people familiar with case control studies in general here? This was a new term for me. I didn't know about that before. No? Okay, so case control studies are a prevalent medicine. Let's say you have a task of, you want to identify whether smoking is related to lung cancer. If you wanted to run a randomized control trial, you could take a group of people, 100 people, randomly divide them into 2 groups of 50 people, and then force one group of people to smoke a lot and the other not to smoke, and then see who gets more lung cancer. Now, that doesn't work very well because you can't force people to smoke, and RCTs are often unavailable in many circumstances. A much cheaper study you can run is a case control study, but it gives you a much lower inference value, but it's still useful. The idea of a case control study is, let's say you want to investigate the relation between lung cancer and smoking. So you take 100 people, 50 people with lung cancer and 50 people without lung cancer. People with cancer will be your cases, and without will be your controls. And then you will count the number of smokers in each of the groups, and then you will draw inference from there. Now, the reason this is a much weaker evidence than randomized control study is because in an RCT, you know for sure that both the control and experimental groups are identical, except for whatever experiment you run in on the experimental group. You have complete control over altering one parameter of the experimental group. So you know you have strong inference. In the case control study, right, you take 100 people, 50 people with cancer, 50 without, and then you count the smokers in each of the groups. But what if you don't know about other parameters which are involved? What if that's, I don't know, blue eye color or orange hair that's involved and has causal relationship to lung cancer, but you never pay attention to those parameters, you have no control over them, and you will never notice. That's why a case control study has lower value, but it's still useful. Okay, so in this particular case, again, a case control study would be us taking EHR records and marking as cases those people that have a particular disease, and then as controls those people without a particular disease. So, yeah, Samar, what's up?
So the data input EHR is at a patient level, right? So we can imagine one patient, every row in the input as one representing one patient EHR, or it could be like EHR, time-based EHR records of different patients. So one record can have one instant of observation of a patient, or just one row of patient with all the observations.
One record is one EHR record. One input is one EHR record for
one EHR record. So one patient can have multiple EHR records.
Oh, is that true?
Yeah.
Okay.
They're having different visits.
Like when you visit, right?
Yeah. Wait, do you generate multiple EHR records when you visit multiple times, or is it a single record that has multiple entries?
So let's say if I visit my doctor for my regular health checkup, so under my account in that service providers, like my hospital, they'll have record for every visit. So every row is a EHR then.
The way I've been reading this paper, and I'm not sure that's correct, was assuming that an EHR maps 1-to-1. A single EHR maps to one-to-one to a single patient.
Yeah, that's where I was also not very clear when I went to whether when we are saying EHR, is it because when the clustering is done, I believe that's at the level of patient.
So patient data, so this is the real data that they talk about. Patient data includes quantitative features consisting of demographic information, diagnosis history, so the entire history, family history, treatment history, vital sign readings, which I assume there's many, medications and laboratory tests. So I think what you mean by a record here is the entire medical history available for a single patient. No,
I think it's at a, because if you take history, right, diagnosis history or lab test history, it's or vital signs, so they change over time, right? So same patient,
yeah. So categorical variables were converted to
exactly,
encoding. Repeated or temporal measurements were encoded as the mean, minimum, maximum count standard.
Oh, okay.
Local record. Does that make sense?
Yeah. Now, so it's essentially they converted a time series to.
Yeah, that's actually a fantastic question. I love this. Okay, that's great.
Like if you have multiple blood pressure measurements, so they're taking the stats of that.
Yeah.
And what I wanted to say was that each time a patient visits a healthcare provider, if their disease status has changed, they can be treated as a new sample. If not, then those aggregation functions like minimum, maximum, mean, whatever, can apply to them. And then eventually they can be converted to categorical.
Yeah, I think the way it is, I mean, from what I could infer is every row is a patient record. So let's say if there are 4 patients and let's just take their vitals as an feature example. And it will be over the period of time you would have captured the vitals multiple times. But to just have one row or one sample representing a patient, you take an average or a mean, and so you just summarize the statistics to have one value and use it to represent your patient.
Yeah, so, you also use the count. You also show the count.
Yeah. So those are counts, and those. So it's essentially a summary of all your very different temporal related or sample related information, and you can converge it into one sample, which is at patient level.
Yeah, this is a good distinction. Thanks for calling this out.
Yeah. Okay.
Thank you.
So in their simulations, they used 4 models. Here so let's read this. Model number 1, the simplest one. Altogether, all relevant. Individuals have the same value, 0, 1, for all hidden input effects. So over here, they visualize the models, and A, B, C are the hidden input effects, right? They generated the clinical data, and then they added a few hidden input effects. So under the model 1, every patient either has all input effects set to 1 or all input effects set to 0. Controls have all hidden effects set to 0. Cases have all hidden effects set to 1. So under this model, if everything is 1, it's a case, otherwise it's a control. Now, why is this useful? A model capturing any hidden input will be able to predict case control status in this scenario, right? So this is sort of like a unit test for your classifier on simulated data. This is not even real data, but then we'll see how these models get more complex and complex. So they're trying to test for different aspects of real EHR data. Okay, model number 2, slightly more complex. An individual have 0 to n hidden input effects chosen at random, right? So this individual had 2 effects set to 1 chosen at random. This one had 1 effect set to 1 chosen at random. One arbitrary effect is used to determine case control status. In controls, this is 0. In cases, this is 1. So here, if the last effect is 1, which could be picked randomly, the case status is 1. If it's 0, the case status, it's a control. A model capturing the relevant hidden input, in this case for these models, will be able to predict case control status in this scenario. This is a test of whether each method can recognize the important hidden effect when there are multiple shifted distributions, right? So in this case, you want to see that a model is able to recognize that the last hidden effect is what matters. Next one, individuals have 0 to n of hidden input effects chosen at random set to 1. The percentage of hidden input effects on represented probability, the percentage of hidden effects set to 1 represents the probability of a patient being a case. A model capturing more hidden effects will be able to more accurately predict case control in this scenario. This is a test of whether each method can perform effectively without a hard rule-based model and could represent a disease with incomplete penetrance. So in this case, oh, sorry, this one is what I should be looking at. 66% of hidden inputs were set to 1. This patient has a 66% chance of being a case. This patient only had 1, and this is at random, only 1 hidden input set to 1, 33% probability of being a case, and they indicated as it being a case. I don't think it's worth going through the 4th model, but is in general the idea of how these different types of simulated data they generated as this idea, does it make sense? We'll see why it's useful and how they're feeding it into the DA in a moment. Yeah, Samar. You're muted.
So essentially, these what the term they use, it's hidden effects. So for every patient record, they add these hidden effects and then randomly switch them on and off to generate more records.
Yeah, in a particular, but not randomly switch them on and off, but according to this set of rules.
Yeah, maybe, or sorry, based on the rules that they have defined. So, and in the first one, they like the first model, they are putting every individuals with the same value. Can we look at the picture in this? Sorry.
Yeah. The picture is much better than the text. Here you go.
Yeah. So in the first model, the hidden effects all are 1. So for every patient who is a case, they'll put just 1 in the input record.
Vice versa, the other way around. So you generate so all this data is simulated. We're not talking about real patient records yet. So you take your simulated patient, then you flip a coin. If it's heads, you set all 3 hidden inputs to 1. If it's tails, you set all 3 hidden inputs to 0. And then depending on that, you call it either a case or control.
Okay. Oh, okay. I thought it's which are the cases?
No. Vice versa. So they generate different types of hidden inputs. And then depending on the hidden inputs, they mark the patient as either a case or a control. And what they want, so what you do is you generate your hidden inputs according to some rules. As a human, you came up with some rules which say, depending on these hidden inputs that you generated, this is either a case or control, maybe in a probabilistic manner.
So what are these hidden inputs? Sorry, I didn't get the essence of a hidden input. What is a hidden input?
It's 3 more. In this case, 3 more parameters in your vector, 3 more.
Yeah, so which means I am in my patient vector, I'm adding 3 more features, which are like A, B, C, right?
Yeah, exactly.
So then if what you're saying is for now to populate those 3 values, what I do is I flip a coin, or based on some rules, I say.
Under model 1, you flip a coin. Under model 2, you do what? I forget.
No, I thought it's the probability one you flip a coin. This is hard coded. The first one is hard coded. You know what are your cases,
So the model case one is all or none. So either you assign all ones or
Yes.
zero.
Yeah,
second
all one.
Second one is of a more of a probabilistic distribution. So within each patient record, you can have a distribution of ABC values, ones and zeros. And the major ones would say whether it's 1 or 0, the case types.
Yeah, but
To me, it looks like all the possible values.
Sorry.
So never mind.
What question are we trying to answer?
So what is the interpretation? I mean, how to interpret these three additional features and
so forgetting, don't look at this table for a moment and just yeah. So you have your patient with a bunch of data, which most of it is irrelevant to prediction of whether the patient is a case or a control. You add a few more variables that are important in predicting whether it's input control or case, and under different models, you know how these hidden effects define whether this is a case or a control, right?
Yeah. So,
and then you want your neural architecture to learn the same heuristics that you came up with. Does that make sense?
So it's like I have my patient data, patient vector. In that, I add some hidden features, which are binary in nature. So in the first setting, model 1 setting, I know whether do I know whether this patient is the case?
No, you don't know, because this is simulated data we're talking about. This is not real patients yet. You're just
so how do I decide, okay, that this guy has to be, or this patient record needs to be a 1 or a 0?
Well, I assume under model 1 you just flip a coin.
Okay, let's see.
Well, it just says 0 or 1. I mean, the best individuals have the same value. The constraint here is that all hidden, under model 1, the constraint is all hidden inputs should be either all 1 or all 0. Right.
Okay. So let's take there are 3 patients for which I have the vectors. So 3 vectors.
Yeah.
I flip a coin I take patient 1, flip a coin. If it comes head, I'll say 1.
Yeah.
Tails 0. And if it comes 1, then I put all the feature values as 1.
Okay.
For AB, right? And for the second one, I flip the coin individually for every feature. Is that
under the second model?
Right.
Yes, that is correct. Yeah.
And then what is the third? Why is it 66.1 and 33.1%?
This one?
Yeah.
Wait, are we finished with model 2? Does it make sense?
Yeah. So it's a random way of doing things, right? So you can do it in one, or you can do it for an individual, binomial or Bernoulli.
Where you decide the case control status based on one hidden feature.
Yes.
Which you know.
Yeah.
But
so if it comes 1 in the last, then you say 1 is case 1. Otherwise, that's fine.
Perfect.
How is the third one?
Yeah, so for the third model, again, you do your, Bernoulli trials, right? You said 1 to 0. And then you look at the aggregate number of ones out of
okay, that way.
With that probability, you generate the case.
Okay. So this is our binomial kind of thing. Okay.
Yeah. The fourth is, I suggest we just skip it. It's in the
same. That's fine. Okay, mainly the idea of how this is being done. That I think I got it.
Okay, cool, great.
So the value of this data augmentation test is to find out if the model is capable of discovering this type of a pattern.
Yep, that is correct. Yeah, the model is able to identify which of these hidden effects are actually the culprits in your disease status. Okay. Oof. So let's if you, this page is very interesting and I think critical here. And then from there it gets easier and then we'll just look at the results effectively. Oh, actually, there is a paragraph which explains how this is related to real medical conditions and why this is realistic, but I'm going to skip it. Okay, if successfully trained, the hidden layer of the denoising autoencoder, remember we trained the DA separately from the, okay, never mind. If successfully trained, the hidden layer of DA, Y, captures the first n factors variation in the data, where n is the number of nodes in the hidden layer, right? Where's the architecture? If the hidden layer is 2, then you capture supposedly 2 most important parameters of your input. Where is it? Where am I reading? To test whether the DA constructed useful features by learning the main factors of variation in data, we use the trained hidden layer as an input to a shallow classifier. To do this, we first completed unsupervised pre-training of the DA with all of the simulated samples, right? You take the simulated samples, you corrupt them, you expect the DA to reconstruct the original input, which it never gets to see because you feed the corrupted input into it. The hidden layers Y were calculated for all samples using a trained DA without any corruption and fed in as the features to a random forest to form a supervised classifier. They use a random forest, whatever, it doesn't matter. Classification. Okay, this is the general idea of what they're trying to do. Does this make sense? I see Samarth is nodding. Okay, cool. The supervised classification comparison was, what is it? And this is, let me think for a second. Yeah, so this in the supervised, okay, so this is the difference between supervised and semi-supervised in their context. In the supervised case, they take all this data and they use it. They take a bunch of patients they simulated. They use it to pre-train the DA for representation. Then they take those values. They don't corrupt them anymore. They input into the DA and then into the classifier. And then because those simulated patients have their case control labels, they train the classifier to predict it. In this case, you have a lot of data to train both the DA and the classifier with the DA. Now, the problem with this is that this is a fully supervised classification algorithm, which doesn't work when you don't have much labeled data, which is the case in real life. The real data is more like the simulated semi-supervised classification situation. The supervised classification comparison was repeated, but with additional patients simulated and utilized during this unsupervised pre-training of the DA. The additional patients were simulated at the same 50% case, 50% control ratio, but their labels were discarded after simulation. Right, so you use a lot of, let's read to Lynn. These additional patients were mixed with the original labeled patients and included in the unsupervised pre-training with DA. The unlabeled samples were then discarded and the DA plus the classifier was then provided the same labeled patient groups as the traditional classifiers. Traditional classifiers are the ones they're comparing against, sort of the baseline classifiers. Labeled patient samples will run through the trained DA in the same manner as the unsupervised pre-training, but without any corruption added to the data. So the point here is, and then they changed this ratio of labeled versus unlabeled data later, and they showed different results. But the point is that you can have a lot of unlabeled data to train the DA, and then very few labeled samples to train both the DA plus a classifier in concert. And the case when the DA plus a classifier outperforms a traditional baseline classifier is when you have very few labeled examples. which is a more realistic case. I know this is confusing. I think it will get clear in a moment. Samar, what's up?
Quickly, so when we say supervised classification comparison, we are assuming that, let's say, we use the same architecture and we have tons of label data, right? And we do classification by training the DA and then using the hidden layer as input to the feature for training, for classification, right?
Yeah, so there's yeah. That's one thing they do. That's the model they're trying to develop, and they are trying to compare its usefulness compared to no DA, just a classifier such as, what is it?
Oh, okay. Fine. So okay, right? so let's say you have a bunch of label data, and you just use that to build the classifier, and then you measure the performance, versus you take both unlabeled and labeled data, build this DA, and then do the classification. Exactly. So that's supervised classification setting.
The second is semi-supervised.
Oh, the second is semi-supervised. Okay, perfect. Yeah. Okay. Just wanted to confirm.
Cool. Any other questions? This is a very confusing part. Like, I reread this like probably 10 times.
No, I think I guessed kind of that's how the encoders are used, autoencoders, but I just wanted to confirm, is there anything specific done different by these guys?
Other folks, anything? I know we're overrunning the time a little bit, but yeah, hopefully we'll be shorter next time. Okay, no questions then. In a moment, I'll show you some graphs now, but before I show them, we need to understand what the graphs are. After you train both DA and the classifier in concert, you can take the hidden layer over here, which is already a reduced dimensionality representation. If the hidden layer is just 2 nodes, then you can represent it immediately in a human readable format in a graph. If it's more nodes, you can run a principal component analysis thing on it to reduce dimensionality even further and show the most principal components to a human, and that's what we're going to see in a second. Okay. And what you ultimately want to see is that after all this training, the PCA should show you the separation, like clustering of cases versus controls patients. Okay. Oof. So we've been talking about the simulated data so far, and this is what I've been talking about. They talk about simulated data for like 9 pages, and then this is this is what they spent talking about real data. So not much. I don't think they actually apply this method much to real data. But the point they're trying to make, the reason it's useful is because the real data set they used is 10,723 patients, but only 3,000 of these patients had known death information. So their task is, where is the task? Let me see. Their task is predict ASL. I don't know what ASL is, but ALS, sorry. Patient survival as the number of days from disease onset. Okay, and they had data on 10,723 patients, but only 3,000 and some of those patients had known death information. So were useful as actual labeled examples. However, thanks to their DA architecture, they were able to perform the unsupervised pre-training part of the DA with all 10,000 patient examples, right? And then the labeled, only 3,000 labeled examples were used for the classifier. That's the point of this entire architecture. Okay. So they show here. Figure 2, where do we reference figure 2? Figure 2, B to F shows the separation of cases from controls as the DA is trained. So as you progress through the controls, as you progress through the epochs, this is your DA separating the controls and cases into the two into the two clusters. So it seems to be working. And then they talk about the fully supervised comparison versus the semi-supervised comparison. Okay, and this is where the big distinction comes from. To examine the ability of DAs to learn the structure of the data, we compare the predictive ability of classification algorithms applied to the DA constructed through unsupervised training. Actually, okay, this paragraph is hard to parse. There is a very good table. In the case of fully supervised training, the DAs didn't offer you much benefit, right? So this is the, is this accuracy? I assume this is accuracy. But basically the DA plus RF, in the case of fully supervised training, didn't offer much benefit over other classifiers, right? You can look at, for 2,000 patients, DA plus RF compared to just RF, there is not much difference. The denoising autoencoder doesn't have much to offer when all labeled data is available to a classifier. Support vector machine performs even better. Samarth, does match your intuition?
Yeah. So essentially what they are saying is, let's say if I had a perfect data set with balanced deaths in two classes, then there's no point in building a DA-based classifier. A regular classifier will definitely be performing better because you're doing it in 2 stages and there is a loss coming in. Like errors in one model, DA model will propagate to your classifier.
Yeah,
so
that's okay. I'm glad this makes sense. This took me such a long time to internalize. Okay, but in the semi-supervised comparison, the situation is different. The full potential of the DA plus RF, again, as a reminder that stands for denoising autoencoder plus random forests, is reflected in semi-supervised parameter sweep comparison for simulation model 1. Each set of parameters, whatever, with sufficient unlabeled examples, the DA method's performance is high, even with very few labeled examples. Because of the extreme feature reduction, the traditional classifier on top of the DA is able to reach its learning capacity with very few labeled examples. This is the results we wanted to achieve. Efficient learning from labeled examples is critical in practical use cases because there are often few well-annotated cases due to the expense of clinical manual review. The 2-node DA plus random forest also showed strong performance in relation to an SVM supported vector machine when there were many observed clinical variables and when there were many hidden effects. The advantages of semi-supervised learning diminishes the number of labeled patients, gets closer to the number of total patients. In addition, at the high patient counts, a DA with more than 2 hidden nodes is required to capture the structure of the data with high resolution. And these are the graphs that show you, so these are the semi-supervised cases, right? And this shows you that when you only have 100, so unsupervised pre-training, only 2-node DA was performed with 10,000 patients. And then these are different cases of how many labeled patients you had. When you only had 100 labeled patients, the DA outperformed by a lot all other classifiers. As you have more labeled examples, the classifiers sort of even out. And the regular classifiers kind of catch up with the DA, which has this benefit of pre-training with 10,000 unlabeled examples. Yeah, I think, okay. This is sort of the idea of this paper. I don't think we need to go through anything else here. Any questions, thoughts, suggestions, feedbacks?
So the high level value of this paper was, like you were saying, wasn't necessarily in their evaluation of the medical data set, but was more about creating this process and building trust in the community about this process and how these tools can be. I mean, this paper was in 2016, right? I think that was in the beginning. Yeah. So this is a while ago. Again, the hardware probably wasn't as expanded by then as it is today. So did Did anybody know if there's a lot of like reference to this paper?
I think it's like 120 or something. I picked the most reference paper from the first chapter or the second chapter, I don't remember.
Nice. Okay. Yeah.
I think the value what I see from this paper, and it kind of makes sense now in hindsight also, is how do you, let's say in a given data set, if you are given with very few labels, like you have a very sparsely labeled data set, then how are you going to approach it in the sense as in how you can build a more reliable model or a better model with very sparsely labeled data?
The answer is reduce dimensionality, as it turned out. Yeah.
Yes, and I don't know, there is another, I'm just thinking in now is once you have built that encoder, autoencoder, that denoising, and you have that hidden representation. So if you plot that into embedding space, every patient, for example, right? So then you can sample also from that vector space, and you can increase your sample size as well. So it's because it has already captured some hidden patterns in terms of clustering, right? If you just look at it from a clustering point of view, you'll find some clusters.
Yeah. Okay. Wait. Say you're right. Can you repeat your idea about sampling? I didn't get that. What did you mean?
So let's assume we had these 10,000 patients, right? So if you ran it through the denoising encoder and you got that hidden representation. So every patient is now, let's say, a two-dimensional vector, right? Now, if I plot it on my two-dimensional plane, then we'll see clusters, right? There could be some clusters which will start appearing.
I don't think you'll see any.
You can see clusters.
We could, but that's not so. Okay, this is actually this. I didn't get this immediately, so I want to say this out loud. The clustering that they visualize over here, it doesn't happen in the DA after you pre-train the DA. The pre-trained DA doesn't know how to distinguish cases versus controls.
Yes.
But I'm just saying this for my.
So in this example, let's assume everything was blue in color. Right? No orange and blue. Then if you look, you could clearly see patterns like, okay, there is some set of data points which are sitting on one corner of the plane, the other set of data points which are sitting on the other corner of the plane. And that itself gives you a lot of insights. Now, if you look at your samples, now labeled samples, and see where they are lying on these planes, you could clearly then try to make a connection. I mean, without building a classifier will be even better. But if I just place now the label points near on this two-dimensional plane and see a k nearest neighbor, let's assume.
But then, interesting. And then you're saying you would be able to infer something about the unlabeled.
Yes, and now if let's say if I do a k nearest neighbor, I can say, oh, I find more samples in this cluster to be in the area of let's say class 1 or class 0, and then you could actually use that representation to bring in more samples.
This is exactly what I wanted to say. When you don't have the labels, you use some clustering to somehow separate the labels. Then on that, you can train other classification things.
Yeah, that makes sense.
Models.
Yeah, and that's actually what the classifier is also doing. I mean, inherently, the classifier that we put, the random forest or any other thing, that is also doing the same thing, but it's more intuitive in terms of, it's more crude, but you see it's more, I would say, without knowing the labels, and then you bring in, it's like, how do I say? It's like you don't tell the answers. It's like test. You don't tell the answers, and say, oh, this is the answer, this is the answer, this cluster might be class 0, this cluster might be class 1. And then when you compare it with your true labels, the very few true labels that you have,
Yeah.
then you can see, oh yes, there is some I'm able to infer.
Okay, yeah, that's a cool idea. I like that.
But this is good. I mean, what I would really go back and read, and this is the simulation strategy they used.
Yeah.
That's something which looks interesting.
Right, so they give you the performance of the GA versus other architectures for these different models, right? And if you want to compare the results, you can do that. Yeah. This is the thing. Anything else?
Just wanted to thank you all. I learned a lot and I really enjoyed this session.
Yeah.
I look forward to having more of these sessions.
Cool. Let me know if you want to present next weekend. I think the floor is still open. Anyone is welcome.
And just one question, just out of curiosity, are you all on-campus students or online students? Only Rahul is on campus, right?
No, I'm not on campus. I'm in Chicago, so I'm a few hours away.
I'm in Europe.
Oh, okay. Oh, cool. I'm in
Europe.
Toronto.
Who is also in Europe? Oh, Akhil, yeah, you're in London. Yeah.
Yes.
Awesome. Okay, it was really nice meeting you all.
Right. See you later. Bye.
Thanks.
See
Bye.
you.
Thanks, Ilya.
Thank you.
Bye,
Bye-bye.
guys.
Thanks.